Good morning, everyone. So what we're going to see now is basically the implementation of what we have seen on the scientific level, at least part of it. And it's been a work that we have been doing for basically the last 8 to 10 years in the framework of the Human Brain Project. Okay, so the Human Brain Project is still accessible even though the project has finished a few months ago in September 2023. It is a live website accessible at humanbrainproject.eu. And as you can see on the right of my slide, there is the main objective of the project since the beginning. So the Human Brain Project should lay the technical foundation for a new model of ICT-based brain research. So during these last 10 years, many groups, more than 100 groups and 300-400 people on average every year have been working to reach this target. And specifically, what we have been doing has been the development of services and tools for allowing the simulation, building of models, data analysis, uh, anything related to the construction of networks that can help us understand the behavior of the brain. And the main outcome, left alone, of course, the publications and the tools and all that is related to the HBP project, the main outcome visible to the public is the EBRAINS research infrastructure. So EBRAINS is Europe's research infrastructure for brain research, and is an ecosystem of tools and services, web applications, and anything related to brain research. So there's a main website that I'm going to open in a few seconds. That is the homepage that you are seeing right now, and it is available at ebrains.eu. And then there are at least a couple of different platforms that are the wiki.ebrains.eu and the knowledge graph in eBrains that are where the collaborative work is done. So if I open, for example, the wiki, this is a website that is structured. It's more a portal, framework where you can do your work and collaborate with your partners. And here you have the possibility to create workspaces that are called collabs, where you can put your data, your models, you can share data with your collaborators, partners, and so on. Okay. And as you can see, it is a wiki page, basically. So it's easy to use because users are kind of used to it. One of the core parts of eBrains is the knowledge graph. So the knowledge graph is basically an interface to all the data, the software, the models that have been built during the HBP and that are continuously added to this database for public use. So as you can see here on the left menu, we have more than 1,000 data sets. So files, zipped files available for users. So not only for, this is public. So you can just reach the search.kg.ebrains.eu. Everything is in the link of my slides. And you have access to all this. You have more than 200 models and so on and so forth. Software packages, web services, and so on. So for example, if I search for Migliore, I have 4 results here for the data set. I have 84 models that are related to the work that we have been doing in Michele's group during these years, and so on. Okay, so it is accessible not only for use, for downloading, let's say, but also for uploading. This means that researchers all over the world can just drop a line to the support, so that the curators of the knowledge graph, and ask them to have their data uploaded to the knowledge graph. Okay, so let's say I am a researcher for anywhere in the world. I have some data that I want other researchers use because I think that it's useful, or because I think some data analysis can be done in a more efficient way. or for whatever reason, you know that research is going more and more towards open access, right? And it's very good. It's a very good point. So you want to put your data there, even though you're not from HPP, you're not from Europe, you can just write them and ask them to upload your data, your models in the knowledge graph. Okay, for data, especially for sensitive data, there's a curation process. So you might be waiting for some time, but in the end, you will have your data there with all the metadata, the link for downloading and all this. Okay, so our main core business in terms of the technological development was actually related to tools that help researchers doing their research. So inside the eBrains.eu website, we have built a section that is called Interactive Workflows and Use Cases, where we put all the available services and web applications and Jupyter notebooks that are just scripts of code for you to launch or test your models, your data analysis tools, and so on. So we grouped everything in a dedicated portal integrated into eBrains that is related to the use case that we work more on and are of basically concerning the cellular level simulation um, scale. So we have been working on single neuron models, small networks, and also big networks at a biophysically detailed level. That's why we call this, the platform cellular level simulation workflows. So if you go to the eBrains website, okay, here you can explore, of course, the website. There are plenty of links and documentations, and of course the origin of all these, all the partners, and so on. But in terms of where the work is done, you have these panels here that are dedicated to different topics. So data, brain atlases, a lot of images about the brain, validation, health research, and so on. And we put ourselves into this section. So the modeling, simulation, and computing. So if you enter this section, you are provided with the list of all the tools, right, that helps you simulate single cells brain regions, right? And if you go on the left part, so the left menu here, the cellular simulation, you are rather soon provided with all these panels. So these are all the tools and with the link to our platform. Okay, so these interactive workflows. So you open these, you launch it, and another tab is open so that you don't lose the path that you have followed on the eBrains website, where our portal is open and you have all the information related to this specific subgroup of tools and services. Okay. So the way we have structured this portal is this one. So you have some general information, and then you have the list of all the use cases that are accessible via this panel that provides a few subsections related to the topic that you want to explore. So for example, if you're interested in trace analysis, so you have electrophysiological traces that you want to analyze, you want to extract some feature from them. Here we have grouped all the tools for doing your data analysis and research or modeling, or at least first step in your modeling workflow. If you are more interested in, for example, morphology analysis, here we have a couple of tools for visualizing the morphologies of different type of neurons and to rotate them to go inside. So it's, of course, we don't have time to show all the tools. We're going to show just a couple of them. But please bear in mind that this is the entrance door for all the tools related to the cellular simulation. Okay, so the first tool that I want to show you uh, is actually inside here. So inside the, sorry. So the single cell modeling, we want to model in a biophysically detailed way. This means all the ion channels, what Michele showed you before. So we take a single neuron. We want to know the most that we can about that neuron. So its morphology, how it behaves when provided with a stimulus, and so on. And then starting from a real trace, we want to model the neuron in order to fine tuning the parameters of our model. Okay. So the first step in these workflows is actually extracting the features, which are the characteristics of electrophysiological traces, from real traces. So we take data from experimentalists that have usually done patch clamp experiments. This means that you have a cell and you put pipettes on the cell and you record all the fluctuation in the membrane voltage. Okay. Once you have the electrophysiological trace, you extract all the features and then you feed these properties into your model. Okay, so we're going to focus on this neurofeature extract tool that allows you to extract the features, and this one, the Hodgkin-Huxley neuron builder, which is the real tool for building your model and optimizing the parameters. Okay, so you open this, as you see, so most of these tools are user-friendly. You can see from the original panel, you can see these icons, and these icons basically explain whether this tool is for everybody. Everybody means that you don't need to know any programming, you don't need to have any expertise in computing related, let's say, operations. Okay, you can just open it and do something, right? Now the point is, if you have never heard about electrophysiological traces, probably this wouldn't tell much to you, right? But still, the tool itself is very user-friendly and in a more general way, let's say point and click. Okay? So you read, you click on something, and something is performed. Some operation is done. Okay? So we have the neurofeature extract presentation page, and then you click on next, and the second page is a page where we have collected a lot of data for you to extract the features you're interested in. So we left alone for the moment all these keywords, but as you can see, they are related to the species where the data come from. So for example, mouse or rats, the structure of the brain, so hippocampus, cerebellum, and so on, and then some other information. So for example, the type of cell, whether they are interneurons, principal cells, and so on. Okay. So I will select just one data set, which is this one. It loads the data and it shows them. It shows them in a very interactive way, which means this is the entire recording for that cell. And this recording has been done through stimulating the cells. So we have the cell, we inject a current, and we record, okay? And this current is more or less strong. Okay? It can be, of course, current is measured in ampere. So with this stimulation, I have this trace. With this stimulation, I have this trace, and so on. Okay? So you can select the stimulation amplitude that you want to have in order to extract the features from the corresponding trace. And when you are happy enough with your selection, just go to the other cell. It's very easy. So you can basically explore the trace. You can even zoom in and out from the trace and see the details. This one, okay. And then in the end, when you're done, you go here and you click on next. Confirm. Okay, now, still very easy to use. You have a point and click interface for selecting the features that you're interested in. There are more than 100 features, and many of them are very, very specific, right? So you really need to be an expert in electrophysiology or a computational neuroscientist in order to understand what they are relating to, right? But some of them are really easy to understand. For example, the spike count. I mean, spike count is how many spikes I have in that trace, right? Or maybe, you know, the first ISI. ISI, this easy, means interspike intervals. Very simple definition. It's the time between the first spike and the second spike. Okay, so you can select a few of them. And in the end, this web application will extract all these features from the traces that you have selected. So I click next. It's computing. And I'm done. And so the results are now available. I can download them. They are zipped in a zip file. They're basically structured as text files in a different extension that is JSON, but they're basically text files. And let's have a look. Yeah. Okay. Okay, let me open. Okay, yes, this one. Okay, and as you can see, it's very also the output is very easy to understand. We have the step, which is the amplitude of the current that we have chosen, and we have the mean of the features that we have selected. Okay, just have a look at this. Okay, so this is just to show you how you are, what you are provided with when you go through this workflow. Okay, but of course, once you get used to the workflows itself, this will become familiar. But just bear in mind that this is just a text file with the mean and the standard deviation, of course, of the features that you have extracted. Now, we have integrated this tool, which is the neurofeature extract, into a bigger tool, which is the Hodgkin Axon Neuron Builder. And this is the tool where we choose a model and optimize the parameters of the model against the data that we have extracted. Right? So this is a more comprehensive workflow. So we have the feature extraction, the model selection, and the parameter optimization. And all these three steps are grouped here in this panel. So this one, we have already seen it. It's the feature extraction. If I click here, I am provided with the same tool that I just showed you. Okay, this one. The second one is the choice of the model. So we provide the user with a list of models that work well, that have been already optimized for a specific neuron, for example, a hippocampal neuron. Okay? But the user might want to take a model that is already good and works well and optimize the model against some other features, for example. Okay? Because, for example, you're an experimentalist, you don't know much about modeling, say, okay, I do experiment on the hippocampus. So I take a model of the hippocampus that is working well, but I inject my data there and optimize the model against my data. Okay? So if you click on choose the model from Model Catalog, which is another platform belonging to the EBRAINS ecosystem for containing all the models, not only of the hippocampus, but many, many regions already built and working from the ensemble of researchers who contributed to. And also this one is free. So if you want to upload your model, you can just do it by writing an email to the support of EBRAINS. And now once I click on choose my model, I am provided with the list of models. Here you have the name. So for example, this model has this long name, but here you can see that it comes, so it relates to a pyramidal cell in CA1 region of the hippocampus, and it's from rat. And here you have the channels that have been implemented in the models related to the cell. So you can just pick up one of those, and it's download it from the model catalog and put it into the web application here. Okay. By the way, as you can see here, this workflow ID is your personal workflow ID. So every time you start a new workflow, you have your own ID, a different ID, but no one else can have yours, right? This is useful because you can start from a previous workflow that you have started and keep on going from there. For example, let's say you don't have time to extract the features, but you have already chosen the model. Okay, you can download it here, save the entire workflow, and then keep it later, tomorrow, and start again from there. Okay. So now we have basically concluded the first two steps. We have extracted the features. What does this mean? So when I download the model, as I just told you, the model is already optimized, so it works well. So here I am provided with the features that I have used for this optimization. Okay, so I download the model and the features are already available there. So you don't have to go through another feature extraction. But of course you can, because as we were saying, you might want to use your data. So in this case, you delete the features that are automatically provided to you and you start again with the feature extraction. Okay, so feature extraction, model choice, and then we want to optimize the parameters. Now, the optimization is based on something that is called genetic algorithm. This means, well, it is related in a way to machine learning, but let's say the blind one. So the kind of machine learning that doesn't know anything about the system, but tries to do what you want, which is tuning the parameters in order to fit some model. Okay? And this genetic algorithm relies on what is called high performance computing, which is not my laptop, not a powerful computer desktop that I might buy on any online website. But these HPC systems are huge facilities where you have many computers, servers, actually. Each of them has many nodes and many cores, which means processors, right? So in order to finalize this parameter optimization, we relied on HPC facilities. So let's say supercomputer facilities, okay, that gave us access. We launch our optimization job on those facilities and we get the results. Okay, so all this has to be in a way configured. Again, this is done very easily through a user-friendly interface. And here you are provided with a list of HPC systems that you might want to use. Now, in order to use the first three, so this everything that is not service account, so DAINT, Cineca, NSG, you need credentials. So this one, the first one, is a supercomputer in Switzerland. The second one in Italy. The third one is in San Diego, in California, the US. So if I click on this, he already knows who I am because when I open the web application, he recognized my browser. So he knew I am Luca. So it doesn't ask me for credentials. Same here. Here, I need to enter my credentials, but I already have an account on NSG. Okay. Now, what happens to students, for example? In order to have space and time on supercomputers, HPC systems, you usually write a project and saying, and this is usually done by PIs, or senior postdocs. So they know what you're doing. They know how much power they need, how many nodes, and so on. So they write a project and they ask for um, resources. Or, for example, in the case of HPP, these supercomputers are part of the project, so you as a partner have a quote on these HPC systems, okay? But a student that wants to try our tools doesn't have this facility, so it doesn't have the computing power to perform this optimization. So in a way, it would be stuck here. So in order to prevent this, because we want as many users as we can actually to use our tools, we created something that is called a service account utility. And put in a very easy way, this means that the only thing that you have to do is to register to EBRAINS. So we have here, we have the EBRAINS website. Okay, here you have the sign in, okay, which is where you might want to enter the entire platform. But if you go down here, you have this link, sign up. So the only thing that you need is an institutional, which means a university, for example, email address. You go here, you create an account with your email address, and you're done. So you don't need anything else in order to use our tools. So once you have this, you can go back to the Hodgkin-Huxley here. You can just select this service account and the default, one of the two default projects, for example, PitState, and configure your optimization parameters. So I don't go inside here. These are related to the genetic algorithm, and these are related to the supercomputer itself. So as I was saying before, how many nodes, how long you want your process to run, and so on. But what's important here is that once you have your eBrains account, you just go here and then you can click apply and run your optimization. There's a message. Oh, okay. Okay, so he logged me out. Okay, now he logged me in again. Okay, you see? So this arrow is just for logging in. So if I go back here and I suppose I click apply, okay, that's done. You see, all these bars are green now, and everything is ready to be launched. So I click on run optimization. No, okay. So if there's something that is not really useful or correct for your simulation, it suggests. So it says, look, you have to choose 12 at least. Okay, so I modified this, and now I launched optimization. Okay, so the job has been successfully submitted, and now I just wait for the result. So for example, I know more or less how long it will take, and a couple of hours later, 3 hours later, the day after, I just come back here and fetch my my jobs. How do I fetch my jobs? Still the same page. You don't have to go through complex, path. Single set simulation run, you fetch the results. Fetch the results of your jobs. I chose the service account. I chose the Swiss one, and I fetch the jobs. So as you can see, the job that I just submitted is running. Okay? and it will end probably in a couple of hours or 3 hours. In the last days, I have submitted other jobs like these ones, and 4 were successful, 1 failed. These were test jobs, so probably maybe I have just to test whether the submission was fine. I just give half an hour, and half an hour is not enough. So this is the reason for this failed job. Otherwise, you have access to successful jobs. Anyway, in any case, you can download the results. And even in this case, you can see why this job hasn't worked. Okay. Now, let's say that I want to see the results of this optimization. I click on download, and now it starts downloading the results. So just a short resume. What have we done? We have extracted features from electrophysiological traces, which means characteristics of the traces, the time between spikes, the number of spikes, the membrane voltage, the average one, for example, and so on. Then we have chosen a model that works already very well and that we want to optimize against other features, or that we might want to simply run again and see how it works. Okay, so feature extraction, model choice, and then we set some parameters for optimizing the model in a supercomputer. So we just configure the parameters, clicked on launch on supercomputer, and just wait. Okay, so very simple workflow, and everything is user friendly. So is accessible without first of all, without programming, without installing software on your machine. So you don't have to install any software package, any library. Just go here and do your job. So what he is doing now, he has fetched the model, the optimized one, is running the model in order for you to see how the model works in real. Remember that this is a model, so we want this to reproduce the activity that we have observed in the experiments. And so it's simulating the model and analyzing the results of the simulation. Okay. The bar is green, so I can go on. Run simulation. Now, part of the EBRAINS main goal is the integration among tools. Okay. Because when you talk about an ecosystem, you want different tools, different services to talk to each other, right? So you don't want separate things. That's why the ensemble of services and web applications and software packages have been designed in order to talk to each other, depending on the topic that you are interested in. And these tools, this neuron as a service that you read here, has been developed at EPFL, and in collaboration with EPFL, we have integrated it in our web application in order to see the results of our simulation. So this is the model that I have just optimized. When I go to start simulation, it's basically running the model. So what you have seen before in Michele's presentation with the provisional stimuli, now you have it. Okay, the stimulus is step current. You have the information here in the parameter. You have a step current, you have delay, T-stop, the amplitude, and so on. Okay. Now, so you have this spike. You can download it as a text file again and perform your analysis. Okay, so and in the end, if you're happy with your model, you can even register it into the model catalog. Again, a web application with web interface for looking at all the models that have been developed from partners and also external users. Okay, so the last portal that I want to show you is a portal. It looks like a website, but it is a portal, which means that it gives access to many services that are behind, okay, the web pages themselves. and is specific for the study of the hippocampus. But there are twin portals that are being constructed for other brain area, for example, the cerebellum or other ones. Okay? And this one is called the hippocampus hub. Okay? So hippocampushub.eu, you are provided with this page. Now, the hippocampus hub is basically structured into two sections. We have an explore section that again has been done mostly by the EPFL in collaboration with our group. This is a very collaborative work where you can have a look at electrophysiological traces, morphologies, and so on. And the part that we have developed ourselves in terms of coding, in terms of software and web application creation, is the build section. The build section, as you can imagine, is a tool that allows you to build your own models. So we have seen in the Hodgkin axon neuron builder that we took a model already fully fledged with all the parameters, with all the channels, all the equations and so on, and we optimized it. Here, you have the opportunity to create your own models. So for the models, you need electrophysiological data, as we have seen. You need the morphology, and you need the equations of the model itself. You can go through all these 3 steps here in a very easy way by, I would say, cherry picking what you're interested in and then optimize from here. So here you have a lot of data that are not only provided by us or by partners of the Human Brain Project, but for example, other platforms like the Neuromorpho platform, which is a very comprehensive platform for morphologies, or the ModelDB, which is a very comprehensive platform for neural models. And the only thing that you have to do here is to select your data. In this case, we are at the morphology page by clicking on the send and adding it to a cart. So the idea here is more or less like online shopping, right? So you enter a website, say Amazon or whatever, and you choose your product and you put it in the cart, right? So I choose this morphology and it is in the cart now. Now I want to choose some electrophysiological data. Choose this one, okay, the second one, and it is added to the cart. Okay, and so on and so forth. So I go to the models page. All the models at the moment comes from ModelDB, which is another, all the references are here, of course. So I don't want to give you many pages to have a look at, but all the references are here, so you can easily reach the direct link. So I have the model and I choose the component of my models, like here. So I just want to draw your attention to this part. So anything, any of my choice is added here. So any file that I choose to add to the cart is added here. And now I am ready to optimize this model that I have created. And of course, the easiest way to do it is with something that we have already built and you have already seen, which is the Hodgkin axon neuron builder. So we have created an interface, an integration between the hippocampus hub, which is where you choose your model components, and the tool that we have built for optimization of these models, so that you can go to one platform, from one platform to the other in a smooth way, effortlessly, I would say. So here I am in the hippocampus, in the Hodgkin axon neuron builder. I just choose the type of model that I want to optimize. Let's say that my choice at the hippocampus hub was for a pyramidal cell. And here I am provided with my choice. So this is the morphology that I have chosen. This is the parameters that I have chosen in the electrophysiology page. And again, I am provided with the same interface. So you see what we have done. We have chosen the model components. We have sent all the model components to the Hodgkin axon neuron builder, and now we are ready for the optimization. Of course, given that, for example, the electrophysiological traces were only chosen, the features are not yet there. That's why this is orange, not yet green. So I go to extract features, and after a few seconds here, I have the feature, the traces that I have chosen on the other portal. Okay, and I can go on. And now the work from now on, the workflows is exactly the same. Okay, so the take-home message here is EBRAINS and the EBRAINS ecosystem offers a lot of tools and services, not only for expert users, but for scientists, researchers, engineers, anyone who works in research and wants to take advantage of something that is already done and is related to the study of the brain, either data analysis or model building and simulation. So please go to the all these platforms that we have shown today. Of course, if you have any questions or doubts, just drop us a line, and we are very happy to answer. Thank you for your attention.